What's up everybody? So in this video we're going to be looking at this crazy cute piglet, but we're also going to be going over the process of meiosis. This video right here is the HL version. So I covered previously the SL version, which is basically very similar, but the HL version just has a few more details which you will see in this video. So basically, where do we even start? So we start with the human body. The human body is made up of billions of cells. You are made up of billions of cells. Now, most of your cells, around 99.9% of your cells, have a nucleus that only has 46 chromosomes. 46 chromosomes, right? That's the normal amount of chromosomes for a human being. Because each different species have different amount of chromosomes. Now, a very small amount of your cells, around 0.0001, only have 23 chromosomes. So these cells include your sex cells, like your gametes. They're called gametes, so your sperm and your egg cell. If you're male, it's your sperm cell. If you're female, it's your egg cell. And again, this egg cell doesn't really look like a legit egg. It looks more like this. So the question is, why do we have this small proportion of cells that only have 23 chromosomes as opposed to the normal 46? The reason is quite simple. Think about reproduction. During reproduction, we're trying to form a normal human being with 46 cells, right? Just like us. Now, in order to do so, we need two cells that have 23 chromosomes, because when they combine together, that will form a cell, a human cell that has 46 chromosomes, the normal amount. And then that cell will duplicate and duplicate to form an entire human being. So that's why we need these cells to have 23, because when the dad donates the sperm and the mom receives the sperm and they combine together and their DNA combines together, that will form a normal 46 cell, a normal 46 chromosome cell, right? So that's why we need these cells of 23 chromosomes. And meiosis that we're learning about in this video is the process by which we form these cells. Because you may ask, how do we form these cells that only have 23 chromosomes? The answer is meiosis. Okay, so in this video, I'm not going to use a very complicated looking cell when I explain the process of meiosis. I'm going to use a cell like this. Normally, a normal cell is very complicated, has a lot of organelles and a lot of things going on, right? But for the sake of meiosis, we don't need all of these organelles. So I'm going to simplify and make a simple cell here that simply has cytoplasm as you can see here, and a huge nucleus. We're amplifying the nucleus. Now normally, like I said, a normal cell has 46 chromosomes inside the nucleus. 46. I'm going to use a cell here with far fewer, only 6. 1, 2, 3, 4, 5, 6, right? Or you can think of it as 3 pairs, right? A normal cell has 23 pairs of chromosomes or 46 total. What do I mean by pair? So each pair, so for example, the green one is one chromosome pair. One of them is from your mom, so you can see here. So I'll make the striped ones as from your mom, just for simplicity, and the simple plain ones as from your dad. So that's why we have total 46, because we have 23 pairs, and each pair has one from your mom and one from your dad. So again, if you understand this process, I'm going to explain this process with only 3 chromosomes. Otherwise, it's going to look hella complicated and confusing. So if you can understand this process with 3 chromosomes, it works the exact same with the rest. Okay, so don't worry if I'm just using 3. It's just for simplicity's sake. So all I want you to know is 23. We're gonna use 3 chromosomes. So let's keep going. By the way, yeah, each chromosome, remember you have 23 pairs. Each chromosome pair, I made here different colors, and the colors just indicate that they have a different purpose. So for example, I'm just making this up, but this chromosome here, for example, could be for physical traits. This one could be for cell structure or something, and this one could be for your immune system, whatever, okay? I'm just making stuff up. So let's now go into the meiosis, the actual process. So meiosis is split up into 2, meiosis I and then meiosis II. So we're going to start here with meiosis I. But before we get into meiosis, there's one step right before meiosis called interphase. So if we start here with a normal cell, think about this cell as having 46 chromosomes or 23 pairs, right? So I'm just going to show 3 chromosomes though, but now this cell, this normal cell, will undergo interphase. This is before meiosis. Interphase is a phase in which we duplicate the DNA. So now the DNA is being turned from 46 chromosomes to 92, or in other words, 23 pairs to 46 pairs. So why are we doing this? Why are we duplicating the DNA? I thought we're trying to make cells that have only 23 chromosomes, but now we're duplicating the DNA. Why are we doing that? That's like the exact opposite of what we're trying to do. Yeah, that may seem confusing, and it's going to make sense real soon why we duplicate it and why we don't just divide. That will come clear just now. So notice, we duplicated the dad's DNA and the mom's chromosome. So we have now double of everything. Instead of having only one from one from dad and one from mom, now we have two from both. So now we enter prophase. So interphase was simply just a duplication of the chromosomes. Now we're entering prophase. I like to think of prophase as pro meaning before, so it's the most early phase, Now, pay attention, it's prophase 1. Okay, it's the first time we're doing prophase. So what happens during prophase? 3 important things. 1, the nuclear membrane, so the membrane surrounding the nucleus, starts to break apart. Why is this? Because we have so much more DNA. We need to make space. If the wall keeps being tight, then there's no space for all this DNA. So the nuclear membrane starts breaking and expanding, okay? Next, we have these microtubule spindles, these little X-shaped things. These start to form. Now, these are going to have a very important function later, and you'll see later. Now, the next part that happens, so these two things happens. Now, after the DNA duplicated, they start aggregating. So all those with the same function will go together. So in other words, the green ones will go together, the blue ones will go together, and the orange ones will go together. So next, something important happens. This is still during prophase, by the way. So next, something called crossing over is going to happen. Crossing over. And I put a sun here because this is the HL part. This is something that SL won't learn a lot of detail about. They just learn about the process, but they don't learn why it matters. So later in this video, I'm going to make a whole page just about crossing over. So for now, we're just going to briefly go over it. So crossing over is a very simple process by which these DNA that have the same function pair up. So the moms and the dads that have been duplicated, they pair up and they will exchange some DNA. What do I mean by that? Look here. Basically, look at these. Look at the blue one. The blue one, the mom's one, is no longer entirely mom. The dad's one is no longer entirely dad. They swapped over some DNA. So some of the DNA from the mom got exchanged with that of the dad's, right? So the mom's DNA is no longer completely mom, and the dad's DNA is no longer completely dad. Now, bear in mind, this doesn't always happen. And again, it happens during prophase, right? So it's before metaphase. This is metaphase. But it happens during this stage. Now, it doesn't need to happen. It can happen, but it doesn't always happen. So sometimes they in fact meet up, but they don't cross over, right? And again, the significance I'll describe more later. Just know it can happen, but it doesn't always happen. So next we go into metaphase. In metaphase, these chromosomes will now line up in the middle. M for middle, it's the middle phase. They line up in the middle by the equator. And now, these microtubule spindles that were forming during prophase will move to the opposite sides. And they will grow the spindle fiber. The spindle fiber is a little rope that will attach to the centromere. We call the center of each chromosome the centromere. So it will attach there and grab on. Okay, that's metaphase. And interesting process here. Again, I put a sun here because we're going to talk about it in this video. It's an HL thing. It's called independent assortment. And we'll get into exactly what that means. So next, we go into anaphase. Anaphase is very simple. Think about Anna as being a girlfriend you had now for a long time, and you guys just stop liking each other for some reason. You're gonna break up. So it's the phase where you break up with Anna. It's an anaphase. So the same way, these chromosome pairs, these homologous chromosomes, are now gonna break up. So we call it homologous chromosomes when they have the same genes on them, but one is from the dad and one is from the mom. So these homologous chromosomes will now separate into the opposite ends of the cell as the spindle fiber starts to shorten. That's anaphase. Now, after anaphase, we get into the next stage, which is what? I'll just delete this. We go into telophase. Telophase, I think of it as telephone phase. It looks like an old-fashioned telephone. So basically what happens during telophase is the nuclear membrane starts reforming around the DNA that's going to opposite sides. So these three are going to this side, and then a nuclear membrane will start forming around it. Same with this one. And then finally, we'll have cytokinesis, which is basically the splitting of these two, of the cell into two to form two separate cells. So this entire process here was meiosis I. Did we achieve? My question is, did we achieve what we wanted to achieve? No. With meiosis, we want to achieve the process of making cells that have half the amount of DNA, that have 23 chromosomes. Do these cells have half the amount of DNA? No, they have the exact same amount as the previous one. Here we had one from the mom, one from the dad for 23 chromosomes. So we had 23 pairs, and this total made 46 chromosomes. We still have the same thing going on here. The only difference is now these chromosomes are not exactly the same as before. So what do I mean by that? Now these chromosomes, due to crossing over, are a bit mixed up. So now this chromosome is mostly dad, but somewhat mom. This one is mostly mom, but somewhat dad. This one is mostly dad, but somewhat mom. So we mixed up the DNA a bit. We really mixed the mom and dad's DNA due to this crossing over, but we still have all the DNA. We still have 46 chromosomes. So we didn't get there yet. So that's what meiosis II is for. So now we're going to go into meiosis II. So each of these cells now will enter meiosis II. So let's take one. Let's take one of them. So we're going to take this one here. This one will now enter meiosis II, even though this happens with both. So just remember it happens with both. So now, basically, we enter again prophase. Only now it's called prophase II. Prophase II. So again, here the nuclear membrane is intact, and now it's going to start disintegrating again. The nuclear envelope starts to disintegrate. These microtubule spindles start forming again, right? Only this time there's no crossing over because there's only one chromosome from each type. There's not two of them, so they can't cross over anymore like here, right? Here we were able to cross them over, but now we're not because there's only one. Because in meiosis, before meiosis II, duplication doesn't happen. Okay, we go straight from the cells that was formed during meiosis I. So now we enter metaphase II. Metaphase, again, very simple, M for middle. They line up right in the equator. The microtubule spindles form the spindle fiber, which will attach to the center of the centromeres. And this time, instead of pulling the homologous chromosomes apart, like here, remember, we pulled homologous chromosomes apart. This time we pull the individual chromatids from this chromosome apart. So we'll pull this one away and this one away. So that we pull the sister chromatids apart. And this process happens in anaphase, right? When these spindle fibers shorten. Then we go into telophase. Again, the nuclear membrane forms around them. And then finally cytokinesis. And now we formed two daughter cells that have how many chromosomes? 23. So this makes sense. We finally did it. We turned it from being here, a 46 chromosome cell, into what? A 23 chromosome cell. We managed to form now sperm and egg. So these here represent sperm and egg. Each of these cells now, basically if sperm is going to be in your testes, and if it's a female, be inside the female, right? So now this sperm can go and when it gets released as sperm, can be combined with an egg cell that also has half the DNA, and that can form a baby cell that's going to have 46 chromosomes, right? So that's pretty much what we just did. Now, unfortunately, that's not where it stops for HL. For HL, we got to learn a little bit more regarding crossing over and independent assortment. So let's go into crossing over. So again, crossing over happens during prophase I. So I have a whole page here, which we'll talk about. Crossing over. So what is crossing over? So again, like I said before, crossing over is when these chromosomes that have been duplicated, so this one is from your mom, this one is from your dad, when they literally cross over. So we call this spot where they cross over the chiasma. So it spells like this, or chiasmata. There's different ways of saying it, but it's chiasma. Now here, when they cross over, what happens is they break off. These legs will break off. So for example, the dad's one will break off and the mom's one will break off. Now instead of these pieces that broke off going back towards their original chromosome and reattaching, they instead attach to the other chromosome. So it will end up looking something like this, right? They basically switch pieces of DNA. Now, This can happen. Again, it doesn't have to happen, but in these cases, it happened for all three of them, but it doesn't have to happen. Now, what does this do? What does crossing over do? What is the purpose of crossing over? So crossing over allows this to happen. It allows our final gametes that were formed to be able to have both DNA from the mom and from the dad. Notice these. They are not, if crossing over did not happen here during meiosis I, during prophase, all of these would be completely mom or completely dad, which means that these final gametes formed would be completely mom or completely dad. But due to this crossing over, we managed to form these chromosomes that are mixed, and that's great because now your sperm contains some information from both your own mom and your own dad, right? Not just your mom or just your dad. So that's great. That's important because this promotes variation. Because now your child will have DNA from both your mom and your dad, not just one of them. So this promotes variation. This promotes uniqueness in a sense. So that's the main purpose of crossing over, to promote variation. Okay, so now the next one, the next HL part we need to know is independent assortment. What is this? So remember, after prophase, when this crossing over happened, during metaphase, they will align in the middle, right? In the middle. M for metaphase, for middle. So when they line up, there's something important to consider. What does independent assortment mean? Independent means you can depend on yourself. You can do what you want. You are independent. You're an independent, strong, independent woman or whatever. Now, that's the same for these chromosomes. They are now independent. They can do what they want. So they can independently sort themselves. So what I mean by this is when these chromosomes cross over and they're going to line up in the middle, they always have to line up next to their own kind. So the green ones have to line up with the green ones, blue ones with the blue ones, and so on. But they can choose which side they line up. So for example, this one here can line up on the left, and this one can line up on the right. And this way, the babies or the daughter cells that will form after meiosis I and meiosis II will all of these three, this one here, and this one here, will move to one cell, and then that will divide to form their own daughter, to form the sex cells. And the other cell will divide and bring these to that side. So what I mean by independent assortment is that it can either be aligned like this, or they can align the opposite way. They can choose how they want to align. Now, depending on how they align, that can produce a different set of outcomes, right? Because in the previous scenario here, let me show you, the baby cell would have had these three chromosomes, this one, and this one, right? But if these two chromosomes align the opposite way, now the baby cell will have a different set of chromosomes. So instead of having this one, it will have this one, and this one. And this means that the final cell, the final gamete formed, will have a different set of chromosomes because this side, the one that forms here after splitting, will now have one of these chromosomes instead of one of these. So independent assortment is another form of promoting uniqueness. And I'll show exactly, I'll try and use some examples. Just like crossing over, independent assortment can promote uniqueness. It can make the gametes as unique as possible. So let me show you a bit more clearly. So pretend, this is still regarding independent assortment. So pretend we just had one chromosome trait. So we just have the blue one. Now these can either align, this one can align on the left and that one can align on the right. But even if they align differently, it's still going to make the same outcome, right? So even if the left one, the dad one goes on the left and that one goes on the right, you're still going to have this outcome. Two of the cells will have the dads and two of the cells will have the mom. You only have two outcomes no matter what, no matter how these two align themselves. Now, what about if we had two traits? So now we're using two colors. What's going to happen? It can either align like this, so the mom's on one side and the dad's on one side, or the mom and the dad, or the dad and the mom, right? Like this. They can align slightly differently. Now, depending on which alignment happens, you'll get four different gametes, or four gametes. So let me show you. So if this happens, then say these two, the moms move to one cell, and then that cell will split. All of those cells that split will get the moms, whereas the other side will get the dads. But due to independent assortment, this possibility is also possible. So now having this arrangement, the daughter cells will be different. Now these two chromosomes here will move to one cell and split. And this will cause the daughter cells, or the gametes, to have this arrangement of chromosomes. So one from the mom and one from the dad. That is different from what happened here, right? So that's interesting. So this shows how aligning randomly, aligning independently, can also promote different possibilities. Because if they always had to align like this, with the mom on one side and the dad on one side, then you can only have two options of children. But because of this independent assortment, like that this can happen or this can happen, now we have four possibilities. So this can happen, this can happen, this can happen, or this can happen, right? Now let's bump it up to even 3 chromosomes to see how many possibilities there is. So if we have 3 unique ones, they can be like this. Maybe it's hard to see, but you got to try and look close. If the quality is good, I'm not sure how good the quality is. But now they can align like this, due to independent assortment. And this means that we can form 8 unique gametes. So this one is unique, this one is unique, this one is unique, this one is unique. 8 of them are unique. Some of them duplicate, but 8 are unique. But if they were not able to arrange themselves uniquely like this, we would only have 2 possibilities. Like say they could only line up like this. Then you'd only be able to form 2 unique gametes. So it's super clear how these gametes, these independent assortment allows the production of completely unique gametes. So let's go back up to here. It allows the production of completely unique gametes, completely unique, many possibilities. That's why you and your sibling look so much different. That's why if your parents have 1,000,000 kids, they will all look slightly different because of this crossing over and independent assortment, making the sperm and the egg cells unique, making the kid that will be produced unique. One last thing here. So we just used 3 pairs, but we know we use 3 chromosomes, but we know that in reality we have 23. So how many possibilities will that bring? The rule to follow is 2 to the power n. Okay, so we saw that if we had n representing the amount of types of chromosomes. So we know with one unique chromosome, like the blue chromosome, the first one we used, the possibilities was 2 unique ones, right? Because there's only one of the dad, one of the mom. The other ones are the same. So 2 to the power of 1, in other words, 2 to the power of 1 is 2. On the contrary, when we had these two different types of chromosomes, the possibility was 4. That's because it was 2 to the power of 2 representing the amount of unique chromosomes. And now again for 3, this one right here, we have 2 to the power of 3. And that gives us 8 unique combinations. Now, if you have 23 chromosomes, which is what we do, the amount of unique gametes you can make due to independent assortment is 2 to the power of 23. Now, that's crazy. That's a massive number of unique gametes that you can make. Now, it's actually the amount of gametes, the unique gametes you can make is much more than this, because we didn't even consider crossing over here. We considered that no crossing over happened. So imagine if we consider crossing over, there'd be so many more possibilities as well. So the combination of crossing over and independent assortment provides a huge variety of unique gametes. So that's the 2 points that you need to understand from meiosis. Meiosis is a process by which we make these cells that have half the amount of DNA, sperm and egg. But also, meiosis promotes genetic diversity through crossing over and independent assortment, so that we can have these sperm and egg cells that are completely unique and make so many different possibilities of children possible.